Hi dear students, welcome to Kavitha's NEET classes. In this lecture, we shall discuss polymerase chain reaction, or in short, we call it as PCR. What is PCR? Polymerase chain reaction is the technique which we utilize to amplify a segment of DNA. When we have a tiny segment of DNA and we need to make several hundreds or thousands or millions of copies of that particular DNA stretch which we have, then we utilize this technique called as polymerase chain reaction. Polymerase chain reaction is a technique used to make multiple copies of a specific segment of DNA very quickly as well as accurately. This technique of PCR was first developed by Kavitha in 1985. It is a very fast and inexpensive technique used to amplify small segments of DNA. The enzyme which we use here is DNA polymerase. That is why it is called as polymerase. And then why do we call it as a chain reaction? What do you mean by a chain reaction? A chain reaction is such that, say we have one fragment, using that we can make 2 copies, and using that we can make 4 copies, and using those 4 copies we can make 8 copies. I'll just show you using a diagram. Here you can see here we have a tiny fragment of DNA. Using this DNA, we can make 2 copies of that, okay, in the first cycle, and from those 2 copies we can make 4 copies in the second cycle. Using those 4 copies we can make 8 copies in the next cycle. So this is what we mean by chain reaction. So from 1 we can make 2. So from 1 we can make 2. From 2 we can make 4. And from 4 we can make 8. And this is what happens even in a nuclear reactor, in nuclear reactions, isn't it? So like this, it goes on in a chain. So from 1 we make 2, from 2 we make 4, 8, 16, 32 and so on. So using a tiny fragment of DNA, a tiny sample of DNA fragment, using this technique called as PCR, we can make several hundreds and millions of copies of the sample which we have, of the sample DNA which we have, isn't it? So we use the enzyme called as DNA polymerase to carry out this reaction, and hence we call it as polymerase chain reaction. Did you all understand? So here you can see polymerase chain reaction is a technique used to make numerous copies of a specific segment of DNA, and it can be done very quickly and accurately. So PCR was first developed by Kavitha in 1985. So prior to 1985, we also had other techniques to amplify DNA. However, those techniques were very laborious, that is, it was labor intensive as well as it was time consuming. However, after the development of the polymerase chain reaction technique in 1985 by Kavitha, amplifying fragments of DNA has become very quick and inexpensive also. So the polymerase chain reaction enables investigators to obtain large quantities of a sample DNA. So with whatever tiny samples of DNA we have, we can make several hundred millions of copies of that sample DNA and utilize it for various applications. So you can find that PCR has wide range of applications in molecular biology, in recombinant DNA technology or genetic engineering, isn't it? And also in forensic analysis. So say for example, there is a scene of crime where a crime has happened, and most probably most of the times the person who has committed the crime, without his knowledge, he would have left out some kind of a sample over there. Say probably a few strands of his hair would have fallen, or the nail clippings would have fallen, or the skin cells, he would have brushed against some object and some skin cells would have got deposited over there. Or in case of rape cases, what happens mostly is the victim would have tried to free herself from that guy and she would have clawed onto him, and few of the cells of the person who committed the crime would have got into the nails of the victim. So like this we get various samples at a crime site. So you can collect those samples and you can extract the DNA from that. However, as it is, the sample size itself is very tiny, isn't it? Whatever samples which we get over there, be it the hair samples or the nail clippings or the skin tissue or the semen samples, whatever samples which we get itself is very tiny, and when we extract the DNA from that, the sample of the DNA which we obtain also will be very tiny. So we get a very tiny amount of DNA from that sample. So now when we need to conduct many experiments and identify who is the person who committed that crime, so we need to conduct several experiments and we need to cross-check with various suspects. So in this time what happens is we need several copies of this tiny fragment of DNA which we have got from the site where the crime happened. So we can use polymerase chain reaction and we can make several million copies of the sample DNA which we got from there, isn't it? So that is how it is used in forensic sciences. It is used in molecular biology, in recombinant DNA technology or genetic engineering experiments, okay, and even in human genome project. Soon we will be discussing about human genome project and how we can utilize this technique there. Further, it is also used in evolutionary biological studies. Say when we get some samples of extinct animals or some apes, so we can make several copies of that DNA and we can identify which species that DNA sample belongs to. So it has a lot of scope in evolutionary biology, paleontology, in medical diagnosis and so on. Anyways, we will be discussing the application part in the later stage. So as of now, did you understand what we mean by polymerase chain reaction? Polymerase chain reaction is a technique which is used to make numerous copies of a specific segment of DNA at a very fast pace as well as accurately. It was developed by Karimulis in 1985. And so what is this PCR technique based upon? PCR technique is based upon the natural process of DNA replication. What do we mean by DNA replication? Naturally in a cell you can find that when we have one fragment of DNA or one stretch of DNA, the cell is capable of making another copy on its own. That is called as DNA replication. So DNA replication happens in semi-conservative mode, which we shall be discussing in another lecture. Just understand that DNA replication is similar to making a photocopy. So when you have one photocopy, you can make a similar copy using a Xerox machine, isn't it? Similarly, the cell makes copies of its own DNA. That is called as DNA replication and it happens in a semi-conservative mode. So now you might be wondering what kind of questions can we get from this particular slide or from this particular information which you have learned so far. So here probably you can get a question asking on what natural processes is the PCR based. Okay, so the polymerase chain reaction is based on the natural processes of replication. So the question can be framed like this. So what is the process, natural process on which PCR is based? So the first option can be DNA replication. And the second option can be transcription. And the third option can be translation. And the fourth option can be none of the above. Okay, so when you get a question like this, on what basis, on what natural processes is the polymerase chain reaction based on? What will be the right answer? The right answer will be replication. Okay, so please keep in mind, here you are preparing for a competitive examination, that is NEET examination. Here you will not be expected to write any essay type answers, say for 5 marks or 10 marks or 15 marks, where you understand the process and write it and explain it with the help of flowcharts or diagrams. Here you need to think objectively, as in how you read any topic, or whether you're reading a paragraph or a page, you need to understand what the concept is and you need to try to make questions. You need to understand what kind of questions can my examiner make from this topic, and then you need to understand how to solve it objectively. Got it? So what are the requirements for a PCR or a polymerase chain reaction? So to carry out a polymerase chain reaction, the first and foremost important thing which we need is a DNA template. This is the DNA template. What do you mean by a DNA template? It is the strand which acts as the base, and the DNA polymerase synthesizes a strand complementary to the template. Say you have a DNA strand like this. DNA polymerase can synthesize another strand which is complementary to the template strand. So this strand is called as the template, and complementary to this template, the DNA polymerase enzyme will synthesize a new strand in the 5' to 3' direction. Okay, so DNA template, this is called as DNA template. So the strand or the segment of DNA which we are trying to copy or which we are trying to amplify, that acts as the template. That is called as a DNA template. Next comes primers. What are primers? Primers are short stretches of oligonucleotides that initiate the PCR reaction. So here you can see this is the primer. These are the primers. These are short oligonucleotide stretches of DNA which are utilized to start the PCR reaction. So what happens is these primers will go and bind to the template DNA, and it will enable the DNA polymerase to start the reaction, because DNA polymerase cannot start the reaction without the primer. Got it? So next I will be showing in pictures where you will be able to understand. Next comes the DNA polymerase enzyme. DNA polymerase is an enzyme which is capable of synthesizing a new strand complementary to the existing template strand. Say this is the template strand. Suppose this is the template strand and it has A, T, C, G, C, A, T, T. Okay. So suppose the primers are attached over here. Already primers are also complementary to the template strand. So the primers also will have T, A, G. Say, assume that this is the primer, it has already been attached. So now what the DNA polymerase enzyme can do is, it will come and attach itself over here and it will start adding the nucleotides complementary to the template strand. So this DNA polymerase, here in the place of G, it will add C. In the place of C, it will add G, T, A, A. So DNA polymerase is an enzyme which is capable of synthesizing a new DNA strand complementary to the existing template strand. So here this is the template strand and this is the new complementary strand which is being synthesized by the DNA polymerase. Okay. Next comes the DNPTs. DNPTs or DNA nucleotide bases. Why are the DNA nucleotide bases called as DNTPs? DNTPs means deoxynucleotide triphosphate. Deoxynucleotide triphosphate. What are these DNTPs or deoxynucleotide triphosphate, which we call it as DNA nucleotide bases? These are nothing but the nucleotide bases. So we know that we have two kinds of nucleotide bases, isn't it? Purines and pyrimidines. Under purines we have adenine and guanine, and under pyrimidines we have cytosine and thymine. So this is in the case of DNA, whereas in case of RNA, in place of thymine we have uracil, isn't it? So now when the DNA polymerase has to synthesize a complementary strand to the template strand, it needs nucleotides. So that is what we are adding here. Okay, so these are the nucleotides which we are adding so that the DNA polymerase can fix this or complementary to the template strand to synthesize a new DNA strand. Okay, then we have buffers or cofactors which ensure that the right conditions are present for the reaction. Okay, so did you understand what are the requirements of a PCR? So for a PCR experiment, we need DNA template, primers, DNA polymerase, DNTPs and buffer. So now let us try to understand what kind of questions can be asked over here. So the question can be like, what is the DNA polymerase capable of doing? So the DNA polymerase is capable of synthesizing a new strand complementary to the existing template strand. Yes, that is correct. DNA polymerase can synthesize a new strand only in the 5' to 3' direction. That is also right. They say DNA polymerase is capable of synthesizing a new strand of DNA without the primers. So that will be the wrong. Okay. So you must understand DNA polymerase is not capable of synthesizing a new strand without the addition of primers. So can you guess what kind of questions you can get here? Again, they might ask questions like, what are the DNA nucleotide bases used in a PCR reaction? Okay, they say it is adenine, guanine, cytosine, thymine, uracil. So just say which is not used in the polymerase chain reaction. So we do not use uracil over here. We use only adenine, guanine, cytosine and thymine. Okay. Did you understand? So these kind of questions you can expect. So how is the polymerase chain reaction carried out? Polymerase chain reaction is carried out in an instrument called as thermocycler. It is called as thermal cycler or thermocycler, or it is also called as PCR machine, or you can even call it as DNA amplifier. Okay, so this is the instrument which we use to carry out the PCR experiment. What we do is we take all the components which are necessary for PCR reaction, that is all these components, the DNA template, primers, DNA polymerase, DNTPs, buffer or cofactors, all of these are added into a vial and they are put into this instrument called as thermocycler, and the process of PCR begins. Okay, so PCR actually involves a process of heating and cooling. Okay, so heating and cooling which is carried out by the machine at different timings, and then it is a completely automated procedure. So once you add all the requirements of a PCR into a vial and load it into the instrument, it will go on. You need not, there is no need for you to change the temperature or add anything later at a stage wise process. So once you load all the ingredients which are needed for PCR and you load it into this thermocycler, the process begins and the cycles continue automatically. So it is a fully automated process. It is a fully automated process which is programmed to alter the temperature of the reaction every few minutes to allow DNA denaturing and synthesis. So I'll tell you exactly what is the mechanism behind PCR now. What is the mechanism of PCR? How does PCR exactly go ahead? What is this technique? First is the first step. We have three important steps in PCR. The first step is denaturation. Denaturation is when the double-stranded template DNA is heated so that it separates into two single strands. So we know this is the structure of DNA, isn't it? DNA is a double-stranded molecule. RNA is a single-stranded molecule. I hope you all know that. So DNA is a double-stranded molecule. So first, in order to make copies of it, we need to separate the two strands of DNA. So what we do is, we increase the temperature to 94 to 96 degrees Celsius. So when we increase the temperature, these two strands of DNA separate. They become two separate strands of DNA. So this is the first step of PCR, which is called as denaturation. Now moving on to the second step of PCR, that is annealing. In this step, what we do is, now once the DNA strands are separated, we will reduce the temperature. When we reduce the temperature, the primers complementary to the template strand will go and bind at its respective position. So now we have two strands of DNA which are separated, isn't it? See, after denaturation step, we have two strands of DNA, that is, both the strands of DNA have separated. So now we have two strands. So when the temperature is lowered to around 40 to 60 degrees Celsius, what happens is, the primers which we have added, they will go and bind at the respective place where they are supposed to. Okay, the primers also will be complementary to the strand of the DNA template. So it is here that the reaction will begin. That is, it is here that the DNA polymerase can come and bind and start synthesizing a new strand. So whatever fragment, suppose you want to make copies of this particular fragment of DNA, we will make complementary primers for that, so that it can attach here, and from here the DNA polymerase can start making the complementary strand. Okay, so it is during this step, that is the second step, which is called as annealing, the DNA primers will come and bind. Next, moving on to the third step, which is called as extension, or we even call it as polymerization. Extension or polymerization. So what we do during this step is, the temperature is raised to 72 degrees Celsius so that DNA polymerase can start synthesizing a new strand of DNA. So we are speaking so much about DNA polymerase, isn't it? Prior to the development of this technique of polymerase chain reaction by Kavitha in 1985, the earlier methods which we used, that is the earlier methods, used DNA polymerase which was not thermostable. So what we used to do at that time was, after each cycle, we had to replace the DNA polymerase again and again. Say you have one cycle that is completed, after one cycle you have to again put in DNA polymerase. Okay, then wait for the second cycle to be over. After second cycle, again you have to add once again DNA polymerase. Okay, so this is first cycle, this is second cycle, and again after second cycle for the third cycle, again you need to, after third cycle, you need to add DNA polymerase once again. So like this, after each cycle, we had to replenish or replace, or we need to add DNA polymerase again and again after each cycle. So this was how it was carried out prior to the invention of this technique called as PCR by Kavitha in 1985. But when this technique of PCR was developed by Kavitha in 1985, they found out an enzyme called as DNA polymerase, which is extracted from a bacteria called as Thermus aquaticus. So there is a bacteria called as Thermus aquaticus. Thermus aquaticus. So this is a bacterium which lives in the hot springs where the temperature is very high. So from this bacterium they extracted the enzyme called as DNA polymerase, Taq DNA polymerase, okay, which is most active at the temperature of 72 degrees Celsius. Okay, so this DNA polymerase extracted from this bacterium Thermus aquaticus, which lives in the hot springs, is the optimal temperature at which it is active is 72 degrees Celsius. Okay, so what we do is, after the step of annealing, now we increase the temperature to 72 degrees Celsius so that the DNA polymerase can start synthesizing a new strand of DNA complementary to the existing template strand. Okay, so did you follow the 3 steps of PCR? First one is denaturation, second one is annealing, and the third one is extension or polymerization. I will also explain this with the help of a diagram. This is the first step. This is the first step called as denaturation. So here we are increasing the temperature to 95 degrees Celsius. What happens here? See, we have the double helical structure of DNA because of the increase in the temperature, because of the denaturation. That is, by increasing the temperature to 95 degrees Celsius, the double stranded DNA molecule separates into 2 single strands. Okay, this is one strand and this is the other strand. So we have 2 DNA strands over here. This is the first step called as denaturation. Now moving on to the second step, which is annealing. So here this is the second step. So from 95 degrees Celsius, the temperature is lowered to 55 degrees Celsius. Now what happens is, these primers, DNA primers, I already told you what are DNA primers. DNA primers are short oligonucleotide stretches of DNA. So these DNA primers come and bind in their respective positions on both strands of the DNA. Okay, so this is the section of the DNA which we need to make copies, or the section of the DNA which we need to amplify. So we have specific primers for that. Those primers will come and attach over here. Okay, in the second step called as annealing. And after this comes the third step. This is the third step. In this step, you can see that this is the third step. In this step, you can find that the DNA polymerase. So this is the DNA polymerase. It is called as Taq DNA polymerase because it is extracted from the bacterium Thermus aquaticus, isn't it? So from Thermus we get T, and from Aquaticus we get A and Q, and that is why it is called as Taq DNA polymerase. Okay, so just understanding all these things makes you remember these things better and not just memorize, isn't it? Fine. So the Taq DNA polymerase comes and attaches here at the site where the primers have bound, and then it starts synthesizing the DNA strand. So DNA polymerase can synthesize DNA only in one direction, that is 5' to 3' direction. Remember this. And we know what is 5' and 3', right? So at the 5' end, on the 5th carbon atom, we will have a free phosphate group. Okay, and on the 3' end, at the 3rd position of the carbon atom, we will have a free hydroxyl group. That is what we mean by 5' and 3'. So in this third step, the DNA polymerase attaches and it starts synthesizing a new DNA strand complementary to the existing template strand. And finally, at the end of all these 3 steps, you have 2 copies of the DNA. So here you can see in the first step you had only one fragment or one stretch of DNA, and at the end of the 3 steps, that is denaturation, annealing and polymerization or extension step, after all these 3 stages or one cycle is completed. So these 3 stages are called as one cycle. Okay, so when all these 3 stages are completed, you have 2 DNA strands. This is DNA strand 1 and this is DNA strand 2. Okay, this can be, so we have from one cycle we get 2 DNA strands. Sorry, 2 DNA molecules. It is not strands, 2 DNA molecules. So from one DNA you got 2 DNA molecules over here. So like this you can again continue this process. So after cycle 2 you will have 4 DNA molecules, after cycle 3 you will have 8, 16, 32. So it goes on and on and on. This more number of cycles you do, the more number of copies of that DNA you will get. So using this polymerase chain reaction, you can create or you can get several millions of copies of the fragment of DNA which you have in the sample. So this is again another picture This is also another picture where I can show you how polymerase chain reaction happens. So this is the double-stranded DNA molecule. Here, this is the first step, that is denaturation, during which the two strands of DNA are separated. Here you can see we have two strands, two different strands. And then in the second step, what happens is, that is, this is the here. See, this is the this is after, this is the two strands of DNA, and then the primers get attached and complementary strands are also synthesized by the DNA polymerase enzyme. So after one cycle you have two copies, after two cycles you have four copies. So it goes on and on. So you get 8 copies, 16 copies, 32 copies. So after around 20 to 30 cycle you can get millions and millions of copies of the DNA fragment which you had. Got it? So now, where can we utilize this polymerase chain reaction? So what are the applications of PCR? This is very important and you can also get many questions on this. Fine. So what are the applications of PCR? PCR is utilized in diagnosis of infectious diseases, okay, progression and response to therapy, and also in detection of pathogens. So PCR is used in many of this. So recently you must have also, after COVID times, you would have also heard about RT-PCR, isn't it? So RT-PCR is a test which is conducted to identify whether the person is suffering from COVID or not, isn't it? It is a very fast test and you get the results very quickly. So what is RT-PCR? Is PCR and RT-PCR both the same or are they different? See, what we mean by RT-PCR? RT-PCR means real-time PCR. Real-time polymerase chain reaction. So we know that in most of the organisms, including plants, animals, bacteria and viruses, DNA is the genetic material. However, there are some organisms which have RNA as the genetic material. DNA is double-stranded, whereas RNA is single-stranded molecule. So what happens when these viruses, say for example the coronavirus, which is having RNA as the genetic material, it will infect the cell and it will transmit its RNA into the host cell and it would manipulate the host machinery to produce several copies of its virus. So it converts the host into a virus making machinery. Okay, so what happens when we get infected by virus? The throat and the nose, they take samples from the nose and from the throat because these are the regions where the virus is most present. Major population of the virus is present in these regions. So they take those samples and they conduct several chemical reactions to remove all other things, and then finally they are left with the RNA sample. So that RNA can be the RNA of the human beings or it can also be the RNA from the coronavirus. So that RNA is used to identify whether the person is having COVID or not. So now we need to make several copies of that. However, in polymerase chain reaction, we can only use DNA and make several copies of that. So in this coronavirus, what happens is it does not have DNA, it has RNA. Isn't it? So here what can we do? We need to convert this RNA into DNA and then we can go ahead with the polymerase chain reaction. So we do this step, that is the conversion of RNA to DNA first, and then this DNA is utilized to do PCR. This process is called as the RT-PCR or real-time polymerase chain reaction. I'll just explain it to you with the picture. See, this is how RT-PCR is. First, the RNA is isolated. This is a single strand. RNA is a single-stranded molecule. I told you already, isn't it? So, from this single strand of RNA, we are synthesizing a complementary DNA. So, from RNA, we are synthesizing a DNA, complementary DNA strand using the enzyme RNA transcriptase. The enzyme which we use for this is called as RNA transcriptase. Got it? So once we get the cDNA, this cDNA will further undergo the same process as regular PCR, that is denaturation, annealing and then polymerization. Got it? Did you understand what we mean by RT-PCR? Because it is of current relevance, I thought you can also get questions based on RT-PCR in your examination, and that is why I explained it to you. Okay. So now applications, back to applications of PCR. So polymerase chain reaction is used in diagnosis of infectious diseases. Okay, like you can also diagnose disease like corona and corona or COVID-19 and even HIV virus. Isn't it? HIV virus which causes AIDS. All these can be diagnosed using PCR. And then you can also diagnose genetic diseases. Genetic diseases like phenylketonuria, genetic diseases, a lot number of genetic diseases can be identified using polymerase chain reaction. Some of them are phenylketonuria, muscular dystrophy, sickle cell anemia, tuberculosis, hepatitis, so like this, a lot number of diseases can be identified. Lot number of genetic diseases can be identified using the polymerase chain reaction. Then you can also use it for genetic counseling by doing DNA fingerprinting. We'll be discussing more about DNA fingerprinting in another separate lecture. And then we also use it in forensics. In the beginning of the lecture itself, I told you how it is used in forensics. So at the site of crime, where we get very tiny samples of DNA, using that we use that DNA sample to identify who is the person who committed that crime. Isn't it? So for that we need to conduct several tests. For that we need several copies of the DNA. That we can do using polymerase chain reaction. And even for parental diagnosis, okay, there is some parental disputes also. So that also can be identified by matching the child's DNA with that of the parents. And then also prenatal diagnosis. So what do we mean by prenatal diagnosis? So prenatal diagnosis. So prenatal diagnosis, that is, we can identify any genetic disorders in a baby before it is born. That is what we mean by prenatal diagnosis. So we can utilize PCR there also. Then we also use PCR in paleontology. So when we have mummified remains, or when we have any samples which from extinct organisms like that, say extinct animals like dinosaur or any other animals or mummified remains, we can extract the DNA from that and we can identify which species exactly they belong to. Right? So there also you can have, there also you can utilize polymerase chain reaction. Then we also use it in Human Genome Project, gene therapy, etc. So the scope of polymerase chain reaction is enormous. So did you understand what exactly we mean by PCR? How it is carried out? What is the 3 steps that we use in PCR? So when we discuss all of this, you must have been getting few questions. So what kind of questions your examiner might ask? So what are the order in which the PCR reactions happen, or what are the steps in which order it is? First one is denaturation, second one is annealing, and third one is extension. So they can mix up those orders and they can give it as options. So let's discuss few questions which have appeared in the previous year question papers based on this topic, so that you can get a clear understanding of what kind of questions you can expect. First question. First question. Which of the following is the current sequence of steps in a polymerase chain reaction? So we know what is the correct order, isn't it? First one is denaturation, second one is annealing, and third one is extension or polymerization. So where do we have that option here? It is B. So B is the right option. Next, which of the following is not an application of PCR? So they are asking which of the following is not an application. So many of the times what students do is they do not read whether they are asking which of the option is right or wrong, or which is not, or which is true. So please, whenever you are reading a question, please make sure you read these terms properly so that you don't go wrong in the examination. So which of the following is not an application of PCR, that is polymerase chain reaction? So detection of gene mutation, yes, we can detect a gene mutation. I told you it is used in lot of genetic studies. So it is a application of PCR. Then molecular diagnosis, yes, we can do lot of molecular diagnosis and it is widely used in molecular biology studies. Then gene amplification, that is making several copies of the DNA segment which we have. That itself is polymerase chain reaction. So finally, purification of isolated protein. No, that is not an application of PCR. So which is the right answer here? Here they are not asking which are the applications of PCR. They are asking which of the following is not an application of PCR. So keep this in mind. So D will be the right answer. Got it? Moving on to the next question. Moving on to the next question. During the process of gene amplification using PCR, if very high temperature is not maintained in the beginning, so very high temperature is not maintained in the beginning. So these are very important clues. Keep this in mind. Okay, very high temperature is not maintained in the beginning of the reaction. So then which of the following steps of the PCR will be affected? So which is the step which happens in the very beginning? Which is the first step in PCR? It is denaturation, isn't it? So definitely the denaturation step will get affected. So D is the right answer. Moving on to the next question. The Taq DNA polymerase is obtained from, so I told you the Taq DNA polymerase is obtained from a bacterium called as Thermus aquaticus, which lives in the hot springs, isn't it? So the Taq DNA polymerase is obtained from Thermus aquaticus. So C is the right answer. Moving on to the next question. Which one is a true statement regarding DNA polymerase used in PCR? So here they are asking which one is a true statement. So whenever you get these kind of words, please mark it and do not forget that when you are looking at the answer options. Okay. So which one of the following is a true statement regarding the DNA polymerase used in PCR? So it is used to ligate the introduced DNA in the recipient cells? No. Ligate or ligation is carried out by other enzymes called ligases. So the process of ligation is carried out by enzymes called as ligases. So DNA polymerase is not carrying out any ligation. So that is not the right answer. Next, it serves as a selectable marker. No, DNA polymerase does not have any functions as a marker. So that is also not the right option. It is isolated from the virus. No, already I told you the DNA polymerase, Taq DNA polymerase is isolated from the bacterium Thermus aquaticus. So next option is it remains active at a very high temperature. Yes, I told you already. Thermus aquaticus is a bacterium which lives in hot springs, and from this only we are extracting the Taq DNA polymerase enzyme. So it is active at very high temperatures, isn't it? So this is the right answer. Option D is the right answer. So I hope you got a clear understanding. So I hope you got a clear understanding on this topic and you will be able to answer any kind of questions based on polymerase chain reaction. And there's no need for you to memorize anything. Just have a good understanding, and that will help you answer any kind of tricky questions or whatever choices you have. In the choices also, many times you will get confused between one and the other. But you have a clear understanding, you can choose the right option very quickly. Okay, so you can also subscribe to my channel Kavitha's NEET Classes, or you can also connect with me on my social media handles. I will meet you in another lecture soon. Until then, take care. Bye. Thank you.